All right. Hello, everyone. Welcome to discussion number 5. So today we are going over the central nervous system, but mainly the sensory system. So we're going to look at our 5 senses, how we physiologically perceive them, and the mechanisms that we use to do so. So I'm going to go ahead and share my screen with you. I will be teaching this discussion through PowerPoint, but I will answer all of the discussion questions via the PowerPoint. So go ahead and fill it along with me. All right, let's go ahead and begin. So again, this is discussion 5, CNS and the sensory system. So question 1 is asking us to explain the structural differences of the 3 types of sensory receptors we have throughout our body, and which of our 5 senses uses each. So let's do a quick refresher and think of what our 5 senses are. So think back and try and remember if you can think of our 5 senses. All right, so our 5 senses are touch, smell, taste, sight, and hearing. So what we're going to look at today is how we perceive these senses and the mechanisms that we use to do so. So we have 3 types of sensory receptors that will pick up these senses. Senses are stimuli, and we have 3 receptors that will pick up on them. First, we have our simple receptors, as well as our complex receptors, and then we have our special senses receptors. So what we're going to see is our simple and complex receptors work together for one of our 5 senses, and the special sense receptors work individually for our 4 other senses. So let's look at these in depth, talk about what they do and their structural differences. So again, our simple and complex go together as they both work throughout the somatosensory system. So our simple and complex receptors work in our somatosensory system, which deals with touch. So our simple and complex receptors have differing functions when it comes to perceiving touch. Our simple receptors detect pain, whereas our complex receptors detect light pressure. So I know that might sound a little complicated because you would think pain is more complex than light touch. However, it's complicated for us, and we need to understand that simple deals with pain and complex deals with light pressure. So let's go ahead and look at the structural differences of these 2 receptors. So here you can see our simple receptor, and here is our complex receptor. If you immediately look at this picture, I'm assuming that the first thing you notice is that the simple receptor is unmyelinated. So our simple receptors are unmyelinated, whereas our complex receptors are myelinated. To further explain this, again, our simple receptors detect pain. And if you have ever stubbed your toe before, for me personally, I stub my toe and then I have a brief moment where I'm like, oh, this is gonna hurt so bad, and then it hurts. Whenever I get my eyebrows waxed, they pull it off and I'm like, this is gonna hurt so bad, and then it hurts. That receptor is unmyelinated, so the signal takes just a little bit longer to actually be sent down the axon and release those neurotransmitters for pain. So that's why pain signals take a brief second to actually be interpreted by the brain. Compared to our complex receptors, which deal with touch, you feel touch immediately. So if someone touches your arm or lightly squeezes your arm or anything, you feel that immediately. Compared to pain, there's always kind of a little moment, a very slight moment before you actually percept and feel the pain itself. And that's due to the unmyelination and the slower impulse travel of the along the axon. All right. One other thing I wanted to point out structural wise about these receptors is they only have one cell. So here is one neuron, one neuron. So the simple receptor is a neuron, as well as the complex receptor, and there's just one cell there. So now let's move on, or never mind. I guess I wanted to talk about the simple receptors and just give you a little bit more of an understanding. So if you've ever seen a diagram of the skin, if you've taken anatomy before, you most definitely have. This is what it looks like, the integumentary system. We have like our hair follicle and our hair shaft. We have our sweat and oil glands. We have blood vessels and all of that. And we also have nerve endings. So this nerve ending is normally called a free nerve ending in the skin, and that is our simple receptor. So you can see that there with those long dendrites. Here you can see a complex receptor, but again, those free nerve endings are talking about our pain receptors. We have 3 types of pain receptors scattered throughout the skin. We have chemical pain receptors, thermal pain receptors, and mechanical pain receptors. So let's use a kitchen example. Let's say we're cooking a spicy meal in the kitchen, and we're cutting up a really hot jalapeno and it splashes on our skin or like our eye or something. That's going to be chemical pain, that kind of like burning, stinging feeling. Thermal pain would be like if you touched the stove or like grabbed a pan out of the oven with just your hand, that like really hot heat pain. And then mechanical pain would be like if you cut yourself or if you slammed your finger in a cabinet. That would be like the squeezing or breaking, that would be mechanical pain. So those are our 3 pain receptors, which are simple receptors, normally called free nerve endings in the skin. All right, so now we will move on to our special sense receptors. So the simple and complex work for touch, special sense receptors work for our other 4 senses. So what we see is the systems these work through are going to be first the olfactory, which is smell, the gustatory system, taste, the visionary system, sight, and the auditory system, hearing. So each of these systems kind of works through its own type of special sense receptors. We see the olfactory and gustatory systems work with chemical receptors, whereas the visionary system works with rods and cones. We'll look at this in depth today. And the auditory system works through hair cell receptors. So those chemical receptors, everything that you smell is chemicals. So like perfume is made of chemicals, gasoline is made of chemicals. Everything that you smell is chemicals that are going to be dissolved in the olfactory epithelium in the nose and be detected and stimulate our special sense receptor in the olfactory and as well as gustatory system. Everything you eat is made up of chemicals. Even natural things like vegetables are made up of different chemicals, natural chemicals. And therefore, whenever they dissolve on the taste buds, our taste buds have special sense receptors that detect the taste of those specific chemicals. Again, we'll look heavily at rods and cones coming up in an example coming soon. Those work through photoreceptors. The rods and cones are known as photoreceptors. And then we have our hair cell receptors, which this picture is actually showing us a hair cell receptor. And you will look heavily at how we stimulate and detect sound in lecture. Dr. Chang dedicates, I think, a whole lecture to how our hair cell receptors work and how the sense of hearing is stimulated and everything. We're just going to briefly look at it in discussion. Let's go ahead and look at those structural differences before we do all this though. So what we see with our special sense receptors is that we use 2 cells. So we use that sensory receptor, and then we also attach it to a neuron of a sensory neuron. So we have a sensory receptor and a sensory neuron. So the sensory receptor is going to detect a stimuli. So in this case, a sound wave. If it was the rods and cones, it would be color or light, and taste or smell, it would be a chemical. So we'll detect that stimuli. This stimuli, if strong enough, will cause the depolarization of the cell, which, remember, depolarization means the cell's voltage is becoming more positive. So depolarization means the cell is becoming more positive. We depolarize the cell. And when we do that, when we depolarize the cell, we will send we will send a receptor potential down the axon that will release neurotransmitters from the axon terminal to our sensory neuron. So one important thing to see here is that sensory receptors generate receptor potentials, not action potentials. Our sensory neurons will generate action potentials, but not our sensory receptors. They generate receptor potentials. So once they generate that receptor potential and release those neurotransmitters from their axon terminal, those neurotransmitters will be sent to the neuron, the sensory neuron. It will receive those neurotransmitters and, if strong enough, generate an action potential to send to the central nervous system, to send to the brain. So that is kind of our pathway for our special sense receptors. Sensory receptor, neuron, brain. So what we see is that this picture is showing us a hair cell. So here we're looking at a hair cell. You can see those little hairs up here. And hair cells work through mechanically gated ion channels. So if you remember last week, we looked at the 3 types of ion channels. We had mechanically gated ion channels, mechanogated ion channels. We had ligand gated ion channels and voltage gated ion channels. So the hair cells work through mechanogated ion channels. Whenever a sound wave enters the ear and travels through the ear canal and reaches the hairs inside of the ear, it will be mechanically pushed and stretched to cause the opening of a channel on that cell membrane. Therefore, ions can flow into the cell, into the sensory receptors, and we will have that receptor potential. If it's strong enough, that's what I mean by can be graded. If the sound isn't strong enough, it won't hit that threshold and depolarize and cause an action potential or a receptor potential. If it's not strong enough, it'll just go back down and we will have no receptor potential and no action potential. So if it's a weak sound, it won't cause threshold. If it's a normal sound, it will cause the reaching of threshold and we will interpret it. And if it's a really loud sound, we'll increase the strength of that action potential and we'll receive, hey, that's really loud. That's why we can kind of deal with volume. But again, you'll hit this really hard in lecture with Dr. Chang. He dedicates almost a whole lecture to it or two. So that's as far as we're going to go into detecting sound in discussion. So let's go ahead and move on. So what is lateral inhibition and how do we use it to identify location of a stimuli? So whenever you see lateral inhibition and location, I want you to match this up with touch or the somatosensory system. Lateral inhibition and identifying location, what we are doing is increasing the precise contrast of where we are being touched. So first I'm going to give us a definition and then I'm going to give you an example. So if you're not a definitions person, that's okay. I'm not either. I'm going to work through an example to illustrate this after we go through the definition. So the definition for lateral inhibition is that it is a way to increase contrast of a physical stimuli in our somatosensory system. So it's a way to increase contrast of touch. So whenever we are touched in a certain area, we know we're being touched here instead of this entire area. Whenever we're touched in 2 different spots, we know we're being touched in 2 different spots instead of just this one spot in between. We are increasing the contrast of that stimuli. Lateral inhibition and the somatosensory system are very precise in identifying location. With the visual system, we can increase contrast of light and colors, not necessarily location. Same for smell. I mean, you can have a general idea of where smell is coming from, but it's not going to be precise. Taste, definitely not going to be precise, like where the taste is coming from, as well as other sense I'm missing. Lateral inhibition in the somatosensory system is extremely precise in identifying location. It's the only one that really works with precise location identification. All right, so let's go ahead and look at this example. So this is something you'll see in Dr. Chang's lecture, but he moves through it pretty quickly. You can also find this in the textbook, but I wanted to use this again to walk through slowly and add some extra understanding so you fully grasp what lateral inhibition is and how it works. So let's break this figure down. So what we see here is a piece of the skin. So we have the skin and we are poking it lightly with a pin. We're not penetrating the skin, so we're not causing pain. We're just lightly poking it with a pin. What receptors are picking up light pressure or light poking? Those are going to be our complex receptors. Our complex receptors pick up light pressure or light touch. We're not penetrating the skin, so we're not causing pain. That would be our simple receptors, but here we're doing light pressure, light poke. So those are our complex receptors. Sorry about that, got cut off. But again, so we're poking our skin, and what we see is that our complex receptors, our primary sensory neurons, are stimulated. So this dark green area and this dark green neuron is the area most directly stimulated with the highest firing rate. However, we have also stimulated the surrounding areas and the neighboring or peripheral neurons. So we've stimulated all three areas, and we're going to use lateral inhibition to make sure that we only percept the poke coming from this centralized area and not this entire area. So let's walk through how we do that. So again, we poke the skin, and our central neuron as well as the neighboring neurons will pick up this stimuli. So we detect that stimuli, and these have the highest firing rate. That central neuron has the biggest firing rate because it's the one most directly stimulated. However, these two neighboring neurons also fire off neurotransmitters. So those neurotransmitters are released to our secondary neuron. And this is the neuron that is bringing this impulse or this signal or stimuli to our brain. So we detect, hey, we're being touched in this area. But what we see is that the secondary neurons have this important job of inhibiting those neighboring neurons from causing further release of neurotransmitters and the further production of action potentials to the brain. So our primary sensory neurons will release those neurotransmitters to the secondary neurons. So our first two neurons in the pathway are stimulated, but that secondary neuron has three branches of axons, and those peripheral axons are going to work through lateral inhibition to prevent these neighboring neurons from releasing neurotransmitters and causing the action potential of these neighboring neurons, but it won't cause the central inhibition of our central neuron. Forgot to add this here. Those secondary neurons are the ones that relay information to the central nervous system. So the primary neurons pick up that stimuli. The secondary neurons carry that information to the central nervous system. The tertiary neurons are the ones that are actually in the brain. So those are the ones where we actually relay the information in the brain of, hey, we're being touched. So what we see is that central neuron is going to still release those neurotransmitters. However, we are going to use lateral inhibition to inhibit those neighboring neurons from releasing those neurotransmitters, causing an action potential and perception in the brain. So an example I like to use here is the neurotransmitter GABA. So the neurotransmitter GABA opens ligand-gated chloride channels along the dendrites and cell bodies of our neurons. Last week, we looked at a neurotransmitter that opened ligand-gated sodium channels along the dendrites and cell bodies of the neurons, and this made the cell more positive. When the cell becomes more positive, what do we have? What happens? The cell depolarizes, and we have an action potential. However, if we open ligand-gated chloride channels, the cell will become more negative, and therefore we won't reach threshold and we won't have an action potential. We won't depolarize and have an action potential. The cell will just stay at a rested state. Therefore, we don't cause depolarization and an action potential, and we do not release neurotransmitters to the neighboring neuron, which is the tertiary neuron, which exists on the brain. Those aren't stimulated, and therefore we never have an action potential and perceive touch coming from this entire area. Instead, we just perceive touch coming from our central pathway in this dark green stimulated area. So that's how we can use lateral inhibition to increase the contrast or preciseness of touch. Instead of stimulating this entire area, we can have a very exact and precise location identification. All right, awesome. So let's go ahead and move on to question number 3. So now we're going to look at large and small receptive fields, the advantages for each, and which one works with rods and which one works with cones. So here's another figure from your textbook, as well as Dr. Chang's slide that I really like because I think it's a great visual for explaining large and small receptive fields. So let's go ahead and start breaking it down. So this side of the photo is showing a large receptive field, and this side of the photo is showing a small receptive field. When we're talking about receptive fields, we're talking about that secondary neuron because that's the one that's actually going to the brain. So let's start over with this this first example. So what we see is that we have this large receptive field. So here is that receptive field of that secondary neuron. You can see we're being poked by a compass now, which has 2 points about 20 millimeters apart. However, as you can see here, even though we're being poked in 2 points, these neurons converge together, forming a large receptive field with that secondary neuron. So we have high convergence of these primary neurons into this secondary neuron, and therefore we're only stimulating one neuron that's going to the brain. So we do not discriminate between these 2 points. We feel just one poke here. So even though we're being poked in 2 places, the neurons that are being directly poked and the neighboring one being stimulated all converge together into that one secondary neuron, and we only perceive touch or poking coming from one area instead of 2 separate areas. So our large receptive fields, what we think of when we think of them is that they have high convergence and low resolution. High convergence meaning we have multiple neurons converging together into our secondary sensory neuron, the one that's going to the brain, and low resolution. It's not a very precise, stimuli. The signal going to the brain is pretty blurry or broad. We feel it coming from this entire area. So you can think of like a camera. If you increase the resolution of a camera, the image becomes very clear. If you decrease the resolution of the camera of a photo, it becomes very blurry. So that's how our large receptive fields work. They have low resolution. You don't have a very precise, location identification. You feel touch coming from just one entire area instead of 2 different areas because of those highly converging neurons into that secondary neuron. So that's our large receptive field. Now let's look at our small receptive field. So we're being poked in the same distance, but now we're in a different area of the body, a more sensitive area of the body. What we see is that with our small receptive fields, we have a smaller receptive field for the secondary neuron. So we stimulate this area and we have one primary neuron that converges into one secondary neuron. Therefore, these are low convergence and high resolution fields. One neuron converges with the secondary neuron, so low convergence, 1-to-1, but high resolution. We have, a stimuli here and we have a stimuli there. We feel 2 different points in this example. So small receptive fields are very precise in location identification, whereas large receptive fields are very broad. We feel just a general one poke, whereas our small receptive fields, we feel 2 different pokes. So when talking about rods and cones, we think of rods as having large receptive fields and cones as having small receptive fields. So rods are responsible for our peripheral vision. So the periphery, you cannot see, or sorry, you can see with your peripheral vision, but you cannot read. So it's not very clear. You can see that there are words there, you can see motion, but it's not a clear image. Compared to our cones that work with central vision, whenever you direct your vision, center your vision, you can actually read. And that's because cones have small receptive fields, very high resolution, very precise images, but low convergence. So let's go ahead and look at the advantages and disadvantages of each and just where these are in the body and some other facts. So our large receptive fields, again, are those high converging and low resolution areas. And we find this in less sensitive body parts. So like our arms, our backs, and our legs. Compared to our small receptive fields, we find these in our hands and face. So if you've ever had a shot in your face, it's way more painful than getting a shot in like your arm or in your thigh, because we have a lot of nerve endings in our face. We have a lot of nerves in our face and our hands, and therefore we have a lot of sensory receptors that can individually converge into our secondary sensory receptors. And so that's why we have higher resolution in these areas. There's a lot more nerve endings, a lot more primary and secondary nerves or neurons. And so those are smaller receptive areas. The advantages of our large receptive areas is that they're cheap to make. We don't have to dedicate a lot of those primary sensory neurons to that area. We can dedicate, just a few, because a lot of those primary ones, we don't have to dedicate a lot of the secondary sensory neurons to that area, because those primary ones will pick up on that stimuli, all converge together into that secondary neuron. So we can, not dedicate that many secondary neurons. However, in our small receptive areas, we have to have a lot of secondary neurons to meet those primary sensory neurons. That's kind of a disadvantage. However, we do get more detailed and fine reception from those areas. All right, cool. So now we're going to identify the differences between rods and cones. So what we see is that rods deal with our peripheral vision. They work in the periphery of the retina, and I'll show you an anatomical picture of this soon. And the cones deal with central vision. They're located at the fovea centralis on the retina, which is like this little freckle-looking feature on the back of the retina. We have about 120 million rods per retina, whereas we have about 6 million cones per retina. So we have a lot more rods, we have a lot more peripheral vision. We only have a little bit of central vision. Rods deal with black and white vision, whereas cones deal with color vision. Rods deal with motion in dim light, whereas cones deal with fine vision and bright light. And then one last time, rods deal with low resolution and high convergence, and cones work for high resolution and low convergence. So if you can remember resolution or convergence, if you just remember one, just know the other is inverse. The resolution part makes a lot more sense to me, like how the peripheral vision is low resolution, it's blurry. So if I know that it's low resolution, I automatically know it's high convergence. Cones are central vision, very high resolution, very precise and detailed resolution. So I know automatically that's low convergence. So if you can remember resolution or if you can remember convergence, just know the other is inverse. Another little memory trick with cones, they start with C. So cones, C, central vision, C, color. Just another way. All right, so now we're gonna move on to looking at phototransduction in our rods and cones and dark current. So this is gonna be the trickiest part of our discussion worksheet today. You're going to definitely wanna know this backwards and forwards because Dr. Chang really likes this process. It's very complex. It is one of the most complicated cellular physiological processes that we will learn. So definitely please understand this because you will see lots of questions over it on the exam. Before we get into it, I did just want to give you all that anatomical idea. Here's the back of the eye. So if you were to cut the eye open and peel that top layer off the back of the eye, this is the retina. And here you can see the fovea centralis. So this is an area of highly packed cones. The retina contains the rods. There are some cones throughout the retina, but the cones are for our central vision are packed here in the fovea centralis. If we were to take a microscopic view of the retina, this is what we would see. These are our photoreceptors, our rods, and yellow is our cones. We know that because the rods, you can see 1, 2, 3 converge together into one sensory neuron, whereas the cones, it's 1 converges into 1 secondary sensory neuron. So low convergence, high resolution, high convergence, low resolution. So these are our photoreceptors, these secondary neurons and secondary, sorry, I've talked so much today. These secondary neurons in the eye are referred to as the bipolar neurons. And then these are the ganglion cells. The ganglion cells work with the basal ganglia in the brain to help us actually detect vision and know what we are seeing. So let's get into phototransduction and dark current. So what are we looking at here? This is a very complicated figure, but let's go ahead and break it down. So what we're looking at is the inside of a rod. This is the cell body and the axon terminal. Rods and cones are very, very similar anatomically and physiologically, and the only difference really is the convergence. So we can think of this as a rod or a cone in these examples. So again, this is our cell body, and here's our axon terminal. So when working through these problems, there's 3 things that we're going to want to look for. We're going to want to look for if rhodopsin is active or inactive, which in our first example, darkness, we see that rhodopsin is inactive. We want to look for if cGMP is high or low. Whenever it's dark, cGMP is high. And the state of the channel, whether it's depolarized or hyperpolarized. So let's begin. Again, our first example is darkness, then we'll get into light. So in darkness, again, we see that rhodopsin is inactive. When we look at the channels that exist on our rods and cones, we have 2. We have 2 channels that exist on our rods and cones. The first is the CNG channel, the cyclic nucleotide gated channel, and then the potassium leak channel. So we have CNG channels, cyclic nucleotide gated channels, and the potassium leak channels. So again, in darkness, we have inactive rhodopsin, and if rhodopsin is inactive, that means we'll have high levels of cGMP. So those are kind of inverse. Inactive rhodopsin, high cGMP. If we have high levels of cGMP, that will allow us to open our CNG channel. So high levels of cGMP will allow us to open our CNG channel, our cyclic nucleotide gated channel. This will allow for the influx or the inflow of calcium and sodium. Calcium and sodium, we know, exist in higher concentrations outside of the cell. So when we open up that channel, they can now move into the cell with their concentration gradient. We also have this potassium leak channel, and this channel is always going to stay open. Potassium will always kind of be leaking out of the cell, but very slow. The width of the potassium leak channel is very small and thin compared to the CNG channel, which is very wide. So whenever we open this CNG channel, calcium and sodium quickly flow into the cell. And when this happens, the cell will become more positive. So when calcium and sodium flow into the cell becomes more positive, and this will cause the cell to do what? This will cause the cell to depolarize. So the depolarization is going to cause our receptor potential. In the neurons, depolarization causes an action potential, but we're looking at our primary sensory neuron here, our photoreceptor. And so we have a receptor potential due to that rapid influx of sodium and calcium through that cyclic nucleotide gated channel. So the cell depolarizes and we have a receptor potential that travels down the axon, reaches the axon terminal, and we will release inhibitory neurotransmitters. So our rods and cones release neurotransmitters that are referred to as inhibitory, and I'll explain why. So the pathway, as we saw before, was rods or cones, bipolar neurons, and then ganglion cells. So if we increase the firing rate or the release of our inhibitory neurotransmitters, we are going to inhibit the release of our bipolar neurons firing rate or release of neurotransmitters. So an increased firing rate of our inhibitory neurotransmitters from our rods and cones is going to lead to a decreased firing rate from our bipolar neurons and a decreased firing rate from our ganglion cells. If we have a decreased firing rate, we will decrease the release of our neurotransmitters to the point where we have no action potential. So if we decrease firing rate, we do not have an action potential. So therefore, in the absence of light, in darkness, we have no action potentials. Those ganglion cells do not stimulate an action potential. They don't depolarize, and therefore we don't detect seeing anything in our brain. That's why when we're in the dark, we cannot see. Our brain is not capable of seeing in the dark because we aren't having action potentials from our ganglion cells. So, complicated process. I definitely recommend drawing this out multiple times, maybe even making a chart, because Dr. Chang will ask you this in the multiple choice section, the multiple choice section, and maybe even the free response section. And he can start anywhere and he can work forwards or backwards. He could ask you, okay, cGMP is high. So does that mean I'm increasing the release of inhibitory neurotransmitters? Rhodopsin is active. Will I have a receptor potential? Calcium and sodium flow through the cell through what type of channel? And when this happens, does it cause an increased firing rate from our bipolar neurons? He can ask you these types of questions forwards and backwards. So make sure you understand this process. I would probably make a chart that's like dark, light, and then fill in, okay, in the darkness, we have inactive rhodopsin, we have high levels of cGMP, we open our CNG channel, calcium and sodium flow in, we depolarize, receptor potential, release inhibitory neurotransmitters, decrease the firing rate of bipolar neurons, decrease the ganglion cells. In the absence of light, we have no action potential. You could say, okay, we have a receptor potential. Do we have an action potential? No, not with our photoreceptors. So make sure you know this process very, very well. So we've worked through darkness. Now let's go on to light. So again, the three things that we're going to want to look at here are if rhodopsin is inactive or active, if cGMP is high or low, and the state of the channel. So what we see is that here we have light. And when light hits our rhodopsin pigment, which is sometimes called opsin, we call this bleaching. Whenever we bleach rhodopsin, it is activated. So bleaching of opsin or bleaching of rhodopsin causes it to be activated. So light hits opsin, it's bleached, it's activated. This will decrease the release of cGMP, so we'll decrease cGMP levels, and that will cause those CNG channels to stay closed. cGMP is what causes those CNG channels to open. If we decrease the amount of cGMP in our rods and cones, those channels will stay closed, and therefore our positive ions will just bounce off of the cell and remain in the extracellular fluid. However, don't forget, we still have our potassium leak channel, which always stays open. So potassium is always going to be slowly leaking out of the cell. And if we're leaking out one of our positive ions, what is this going to do to the voltage of the cell? It's going to cause the voltage to become more negative. So the cell will become more negative, and when the cell becomes more negative, that will cause the cell to hyperpolarize, so that it will continually become more negative and we will have hyperpolarization. So when the cell hyperpolarizes, it doesn't ever reach threshold, and therefore we have no receptor potential. The cell never reaches threshold. That it has to become more positive and not more negative to reach threshold. So the cell continually becomes more negative. Therefore, we never hit threshold and we never have that receptor potential. Therefore, we decrease the release of our inhibitory neurotransmitters. This picture shows just a few of them. We'll always kind of leak out some neurotransmitters, but we aren't releasing enough to cause anything to really occur. So if we decrease the release of our inhibitory neurotransmitters, we're going to again affect that path. So decreased inhibitory neurotransmitters means an increased firing rate from our bipolar neurons, because that's that inverse relationship. Those bipolar neurons are not being inhibited anymore. And an increased firing rate from our bipolar neurons means an increased firing rate from our ganglion cells. Therefore, in the presence of light, we have an action potential. So in the presence of light, we have action potentials of the ganglion cells, which are going to work with the brain, and we will perceive vision. We will see light, we will see colors. So that's how phototransduction works in light. So again, make sure you know this process very, very well. So here's just one other little way to summarize it. So we have some light hitting the eye. So when light hits the eye, is this going to cause an increase or decrease in our firing rate from our rods or cones? Light hits the eye, this will cause a decreased firing rate of those inhibitory neurotransmitters from the rods or cones. Since they're inhibitory, that means it will cause a what firing rate from our bipolar neurons? It will cause an increased firing rate from our bipolar neurons, and therefore it will cause a increased firing rate from our ganglion cells. Therefore, we will have an action potential from our pathways, our pathways leading to the brain. And so in the presence of light, we have action potentials and we perceive light. All right, so lastly, question number 6. We are being asked to explain how the visual and auditory systems identify location of the stimuli. This will go really quickly because we already went through visual and partly auditory. But what we see is that visual location uses the locations of the receptors on the retina to identify locations of light sources as well as color. Auditory location compares the time difference of the action potentials from the two ears to recalculate the source of sound. So if sound hits this ear first, we'll have action potentials from this side of the brain first, and then this side will come in. But again, you're going to go over that very, very, very in depth with Dr. Chang in lecture, as it is probably the most complex physiological process in our sensory system. So that is all I have for y'all. Thank you so much for watching this. I hope it helped. And I will see y'all next week for discussion number 6, our last discussion before exam 2. So have a great week, and I will see y'all later. Bye.